This set of notes is on cell transport. Permeability is the characteristic of a material which allows substances to move through it. And there are three different ways to think of permeability. Something could be fully permeable, and that means that anything would be able to move through the material. Semi-permeable, also called selectively permeable, which means that only certain things are allowed to move through the material. Or it could be impermeable, and that would be substances that cannot move through the material at all. So cell transport is the movement of substances across the cell membrane. And in terms of permeability, the cell membrane is selectively permeable. And so our cell membrane only allows certain substances to move in and out. A concentration gradient is when there's a difference in the concentration between two locations. So one location is going to have a high concentration and another location will have a low concentration. Concentration gradients play a role when it comes to the different types of transport. So there's two main groups of transport. There's passive transport, and the examples of that are diffusion, osmosis, and facilitated diffusion. And then the other category is active transport, and that would include pumps, endocytosis, and exocytosis. We'll start with passive transport. Passive transport is the movement of a substance down a concentration gradient. When we say down a concentration gradient, we mean from high to low concentration. Think of it like having something at the top of a hill and rolling that down the hill. We're going from a high to a low concentration. The goal with passive transport is equilibrium. And even at equilibrium, there's movement back and forth across the membrane. It's just that there's an equal amount of movement in both directions. And in passive transport, no energy is used. One example of passive transport is diffusion. And diffusion is the passive transport of any substance. So it's a general term, which means that something is going from high to low concentration. And different substances will follow their own concentration gradient. So one substance is unaffected by the concentration gradient of another substance. So, like if you're looking at the food coloring image at the bottom, the red is going to move all on its own, the green is going to move all on its own from their own concentration gradient of high to low concentration. They're unaffected by each other. So here is just an example of diffusion moving from high to low concentration. Another example of passive transport is osmosis. And osmosis is the passive transport of water. So water is given its own category in terms of passive transport. And it still moves from high to low concentration and it still uses no energy. Here's an example that's showing osmosis. And in this case, we've got water and then we've got the solute of sugar. Water is able to move across the membrane, but sugar cannot. So sugar's stuck where it is. To reach equilibrium in terms of concentration, water must move from the left side to the right side. Even though there isn't an equal amount on both sides, this results in an equal concentration of water on both sides. So that's important to remember. When we're at equilibrium, it's not always about having the same amount, but it's having the same concentration. The third type of passive transport is facilitated diffusion. And the term facilitate means to help. So facilitated diffusion is passive transport with the aid or the help of a protein. This could happen because the particle that needs to move might be too large to pass through the phospholipid bilayer on its own. It might have a charge differential, but it's going to use some help. And here's just an example of a protein that's being used in facilitated diffusion. Here's a comparison between regular diffusion and facilitated diffusion. Both are still going from high to low concentration. The difference is facilitated diffusion is going to use a helper protein. One thing that we'll be doing as we talk about transport is solving example problems. And so when solving a transport problem, it's most helpful to draw a picture to help you visualize the situation. Because once you're able to visualize the situation, it's going to be a lot easier to determine what's moving and in what direction. So in drawing a picture, you would draw like a circle to represent what is inside the cell, and then anything outside of the circle would represent what's outside of the cell. So here's problem number one. A typical human body cell contains 2% sugar. If it were placed into a solution containing 7% sugar, which way would the sugar diffuse? So first we need to draw a picture of the cell, which has already been done for you. Next, you want to fill in the information you know from the problem. So the problem told us that there was 2% sugar in the human body cell, and there was 7% sugar in the solution. So now that you have that information on the picture, and remembering that diffusion moves substances from high to low concentration, you should be able to determine which way the sugar will move. You want to look to see where there's a greater amount and where there's a lesser amount, and then know that the movement's going to go from the greater amount to the lesser amount. And you want to show that direction of movement on your diagram by putting an arrow on the picture. So in this case, to reach equilibrium, sugar would need to move into the cell from high concentration of 7% to low concentration of 2%. So there's different types of solutions when we're talking about osmosis and water movement. One type of solution is an isotonic solution. Iso means equal. And so an isotonic solution is a solution that has the same solute concentration as the cell. That means the solute and the water concentrations inside the cell are the same as outside. This means that water's at equilibrium, and so the cell will stay at the same size. This is the type of solution that we want our cells to be in. Next, we have a hypertonic solution. The term hyper means high, and so hypertonic solution has a higher solute concentration than the cell. The solute concentration outside the cell is greater than inside the cell. That means the water concentration will always be the opposite. The water concentration outside the cell is lower than inside the cell. So water moves out and the cell will shrink. Now, if we look at the diagram, we can see all of that information is on the diagram, which makes it easier for us to visualize what's going on. So hypertonic means that the solution has a high amount of solute. So if there's high solute outside in the solution, that means there's low solute in the solution. So you can see that written on the diagram. Anytime we have solute information, water will automatically be opposite. So if there's high solute in the solution, that means there's low water concentration in the solution, which means there's high water concentration inside the cell. So now that we have that information and we know that osmosis will have water go from high to low concentration, we know that this cell is going to lose water. And as water moves out, that cell will shrink. And then the final kind of solution is a hypotonic solution. Hypo means low. So this means that a solution has a lower solute concentration than the cell. The solute concentration outside the cell is less than inside. And so since water concentration will be opposite of the solute concentration, the water concentration outside the cell will be greater than inside the cell. Water will move in and the cell will swell. So again, if we look at the diagram, knowing that hypotonic means a low solute concentration in the solution, we can put that as our solute outside the cell, which means that our solute inside the cell will be high. We know that water will be opposite. And so if we have low solute in the solution, we have high water concentration in the solution. Opposite, we have low water concentration in the cell. And knowing osmosis, we'll have water move from high to low concentration. Water will move into the cell, which will cause the cell to swell. All right, so now we have another problem. And here it says that cells have a very low salt content. If you put a cell in a saltwater solution, what way will the water move? So first, we draw a picture of the cell. Next, we put what information we know from the problem. So our cells have a low salt content, and we're putting it in salt water. And so if we have salt water, it's going to have a high salt content. Next, even though it doesn't tell us water information, it is always opposite the solute concentration. So if we are low salt in the cell, we have a high water concentration. And if we have a high saltwater concentration in the solution, we have a low water concentration in the solution. And remembering that osmosis moves water from high to low concentration, we should be able to determine what way the water will move. And we'll show that by drawing an arrow on the picture. In this case, to reach equilibrium, water would need to move out of the cell. This would cause the cell to shrink. If it's losing water, the cell will shrink. Therefore, the salt water would be a hypertonic solution. The salt water has high solute concentration and it causes the cell to lose water. There's more salt in the solution than in the cell. When this happens to a cell, it's called plasmolysis. Basically, it causes the cell to dehydrate. Problem number 3. Cells have a high water content, but it is not purely water. If you put a cell in pure distilled water, what way will the water move? So we first draw a picture of the cell. Next, we fill in the information. Cells we know have a high water content, but since it's not pure water, that means it's actually going to have a lower water concentration than the pure distilled water, which is pure with no solutes in it. Remembering that osmosis moves water from high to low concentration, we can determine which way the water will move and show that movement by drawing an arrow on our picture. So in this case, to reach equilibrium, water would need to move into the cell. This would cause the cell to swell. Therefore, the distilled water would be a hypotonic solution. There is less solute in the solution than in the cell. We know this because the solute concentration will be opposite the water concentration. And when this happens, it's called cytolysis, because oftentimes, especially when an animal cell absorbs excess water, its cell membrane can't hold it anymore, and the cell membrane can burst, and then the cell would die. So, moving on now to the other type of transport, which is active transport. And active transport is different than passive in that active transport will always use energy. Active transport is the movement of a substance against a concentration gradient from low to high concentration. With active transport, the goal is not equilibrium. Instead, the goal is to concentrate substances inside or outside the cell. In a way, the cell is trying to stockpile it. It's either bringing a lot of it in for storage or it's trying to move excess out. So to have this happen, it's going to use energy. One type of active transport are pumps. Pumps will force substances into or out of the cell against the natural flow. A good way to think of this is sump pumps. Some people end up having their basements flood with water when it's especially wet outside. And houses that are prone to this oftentimes will have a sump pump. And what the job of the sump pump is as water flows into the house, the sump pump is trying to pump it out. So water would be flowing into the house by passive transport. It's just happening because there's a high concentration of water outside and a lower concentration of water inside. So the water is going down the concentration gradient. But the sump pump, which uses energy because you usually have to plug that in, it's going to force that excess water out of the house against the concentration gradient, forcing it back outside where there's already a lot of water. Another type of active transport is endocytosis. The prefix endo means into. And so endocytosis is bringing things into the cell by the creation of vacuoles. So if there's something in the environment that the cell wants, it will cause its cell membrane to fold in around it, pinch off, and create a little vacuole with that substance in it. Pinocytosis is bringing in liquid or fluid into the cell, and phagocytosis is bringing in particles. It's also called cell eating, and that's one way our white blood cells are able to fight some infections. The third type of active transport is exocytosis. The prefix exo means exit, and this is pretty much the opposite of endocytosis. This is removing things from the cell through the fusion of vacuoles with the cell membrane. So if there's something inside the cell that the cell wants to get rid of, it will fuse that vacuole with the cell membrane, open up, and then expel that contents into the space outside the cell. So again, in summary, cell transport, we've got active transport, which is going to require energy. And then we've got passive transport, which won't require energy. It happens automatically or spontaneously. Diffusion, facilitated diffusion, and active transport can be explained in a simpler manner. For diffusion, if, for example, you want to move a stone from a higher place, it will slide down the incline to the lower level. Facilitated diffusion, if you think about moving that stone, but you place it in a wagon instead, it will make it down to the lower level more easily. And then active transport, if you want to move the stone up the hill, it's going to need to be carried like in a vehicle using energy like gasoline. So it's just another way to visualize how these types of transport are different.